Hi class, it's me Mr. Morris, and we're going to continue our notes. This is unit 3.9, and we're continuing talking about chromatography. Chromatography is a physical method of separation that distributes components between two phases. There's a stationary phase and a mobile phase. So the mobile phase is a component that moves, and it moves in a definite direction. The stationary phase, that would be normally a solid. It's the substance that is fixed in place for the chromatography procedure. The way that chromatography works is it takes advantage of the different strength of intermolecular forces between the components in the mobile phase, that's the solvent, and between also the other intermolecular forces between the components and the stationary phase. So if it's more attracted, if a component is more attracted to the stationary phase, it will not move with the solvent. It will get stuck, so to speak, in the stationary phase because it's more attracted to the stationary phase than to the mobile phase. If the intermolecular forces are more similar to the mobile phase, then it will flow with the mobile phase right past the stationary phase. Let's go into a little more detail. Paper chromatography is one example. Paper chromatography is a technique that involves placing a small dot or line of sample solution onto a strip of chromatography paper. Then the paper is placed in a container with a shallow layer of solvent and it's sealed. As the solvent rises through the paper, it meets the sample mixture, which starts to travel up, so the sample mixture starts to travel up the paper with the solvent. So the component that's most soluble in the solvent will move up the paper. Okay, then we have TLC, or thin layer chromatography. It's similar to paper chromatography, but instead of using the stationary phase of paper, it involves a stationary phase of a thin layer of an adsorbent like silica gel, alumina, or cellulose on flat, inert substrate. Compared to paper, it has the advantage of being faster, better separations, better quantitative analysis, and the choice between different adsorbents. Then we have column chromatography. Column chromatography is a separation technique in which the liquid, the mobile phase, passes through a column that is packed with stationary phase composed of irregularly or spherically shaped particles, or a porous monolithic layer, or a porous membrane. So it can be any of those things. A lot of times it's something like a burette that has glass beads in it, or something along those lines. So we've got paper chromatography on the left. We have thin layer chromatography being discussed over on the right side a little bit. And then to the far right, this is an example of column chromatography. Let's talk about the general concept and some examples that might be similar to questions you need to be able to answer. The component that has stronger attractions to the stationary phase, the component that has stronger attraction to the stationary phase will be the one with the strongest intermolecular forces with the stationary phase. So what that means is it's going to have similar IMFs. So the stationary phase and that component will have the most similar IMFs. So on the other hand, the component that has a stronger attraction to the mobile phase will be the one with the strongest intermolecular forces with the mobile phase, which means that it's going to have similar IMFs to the mobile phase. Here's an example. In a paper chromatography experiment, a sample of a pigment is separated into two components. We have X and Y, as shown in the figure above. The surface of the paper is moderately polar, so the paper is moderately polar. What could be concluded about X and Y based on the experimental results? So first of all, if the paper is moderately polar, and then we've got hexane as the liquid at the bottom, you can see this hexane here. One thing you need to know about hexane is that it is nonpolar. And the direction of flow of hexane is going to be up the paper. So that's the direction of flow that we have. The mobile phase is moving up the paper. We've got that little dot of ink, or the pigment, and it is going to be carried up the paper at a different rate. So what we want to do is decide whether components X and Y are more polar or non-polar. The one that is more polar is going to be more attracted to the paper than it is the mobile phase. The one that is non-polar is going to be more attracted to the mobile phase, and it will move farther. So what we can do, looking at these options, we can identify A, X has a larger molar mass than Y does. Well, chromatography tells us nothing about molar mass. Y has a larger molar mass than X does. That's not the case necessarily. X is more polar than Y, or Y is more polar than X. Well, Y is the one that didn't go as far, so that would make it more polar. Let's look at another example. A student investigates various dyes using paper chromatography. The student has samples of 3 pure dyes labeled A, B, and C. So we have A, B, and C in this diagram, right here. And an unknown sample that contains one of the 3. We don't know what it is. The student prepares the chromatography chambers shown above. On the left, putting a drop of each dye at the indicated position on the chromatography paper, a polar molecule or a polar material, and standing the paper in a nonpolar solvent. So we've got a nonpolar stationary, or a nonpolar mobile phase again. The developed chromatographs are shown above on the right. So which dye is the least polar? That's what we want to start with. Which one's the least polar? That's going to be the one that went the farthest, since our solvent is nonpolar. So part A is going to be the dye that traveled the farthest, again because it's a nonpolar solvent, a nonpolar mobile phase. So that would be dye C, because it moved the farthest, and the mobile phase is nonpolar. So the least polar component is more strongly attracted to the mobile phase. We could also mention that they have similar LDF, or similar intermolecular forces, because they both have LDFs, being nonpolar molecules. Okay, part B We want to answer that. It says, which dye is present in the unknown sample? So let's compare the knowns and the unknown. Pay attention to where the solvent front is. So you see that the solvent front is farther on the one on the left than it is on the one on the right. So we're looking at what fraction of the solvent front the unknown moved. So the unknown moved about halfway, to the solvent front. So we want to find the one on the dye that moved about halfway to the solvent front, and it looks to me like that would be this dye. So the unknown appears to be dye A, because dye A and the unknown both traveled about half as far as the solvent.